Hello students, in today's lecture we are going to discuss about the ABC flowering model with previous year questions, and all of the information, notes and questions will be available for download from my Telegram channel. Let's move ahead. So in today's lecture we'll discuss about a flower. The first question that we have in front of us is what is a flower? So a flower is basically a modified shoot. The next question that comes to our mind is why it is considered as a modified shoot. It is considered as a modified shoot because it contains condensed nodes and internodes. So nodes and internodes are actually the properties of a shoot or a stem, and since they are present in a flower, therefore a flower is a modified shoot. Moving ahead, the next important thing is to understand what are different parts or whorls of a specific flower. From outside to inside, the whorls of a flower are as follows. The outermost whorl is known as calyx, the unit for which is known as a sepal. The innermost whorl, or the whorl inner to the whorl number 1, is called as corolla. The unit is petal. The next whorl is known as androecium. The unit is known as stamen. And the innermost whorl is known as gynoecium, the unit for which is carpel or pistil. Now, the flower is a derived product of the shoot, and within the shoot, that too a very important part which is known as shoot apical meristem. So let's understand about the shoot apical meristem. Now the shoot apical meristem is a part which is located within a part known as apical bud of a particular plant. So this is an apical bud containing the SAM. SAM is the short form for shoot apical meristem. Within the SAM, if you see that this green structure which has emerged is an enlarged view of the SAM. So the topmost part of the plant that was represented on the left hand corner, this is basically an apical bud. Within the apical bud is present the SAM. So this is basically an enlarged view of the SAM or the shoot apical meristem. Now the thing is, the SAM is full of n number of cells which are undifferentiated. So this is a keyword which everybody should know, that the SAM is containing a lot of undifferentiated cells. By meaning of undifferentiated, we mean that these cells do not know that what they are going to form in future. They need certain signals to know that they will form a part of the shoot or the root system. So yet they do not know anything, but depending upon the signals coming from the surrounding cells or from within them, they are going to form certain type of cells, right? So that process you must have learned is known as differentiation. Upon differentiation, the cells of the shoot apical meristem, they can form anything which can be a leaf, a flower, or even a fruit. Now, the parts of a plant just represented like by a leaf, these are something known as the vegetative portion, or this kind of thing is known as the vegetative parts, right? And something which is of this sort, the flower and the fruit, eventually they are kind of the reproductive units of a particular plant that we are saying. I'm not saying of a flower reproductive whorls, but I'm saying the term vegetative and the term reproductive. I hope that makes sense to everyone over here, right? Now, moving ahead, the next thing that we have in our mind is to understand how a flower actually develops within a SAM or the shoot apical meristem. So, the next important thing that we have is something called as floral development. Now, there are two phases in the life cycle of a plant. One is known as the vegetative phase, where mainly the organs like leaf and other vegetative part are a main focus. And the other phase is known as the reproductive phase. The reproductive phase is mainly associated with formation of flowers and fruits, etc. Now, as you can see, that there's a very important term called as floral induction, right? So floral induction. Now, what is floral induction? So basically, floral induction is a process. It is basically a process which commits the SAM to develop into a flower, which commits a SAM or a shoot apical meristem to develop into a flower. Now, this is very important, right? So Floral induction is basically a process by which shoot apical meristem will turn into a flower. And that event involves a lot of genes, basically two class of genes for which we are going to discuss in this lecture today. Now the thing is, as you can see that the shoot apical meristem can also be of several types, right? Or it can have several types of identity. So the shoot apical meristem can either be vegetative in nature, then you call it as vegetative meristem. Sometimes it will develop into something called as inflorescence meristem, and sometimes it will develop into something called as floral meristem. And the floral meristem is eventually going to result in the formation of a flower. So this is quite an important chain of event that we have to understand over here. I hope that makes sense to everyone, right? Now, we have clearly seen that the vegetative meristem or the vegetative shoot apical meristem is a part of the vegetative phase of the development, while the reproductive phase results in two identities or two types of SAM. One is called as inflorescence meristem and floral meristem. If I discuss in the life cycle of a plant, the first type of SAM is always vegetative. Then it results in the formation of something called as inflorescence. Then it results in the formation of something called as floral meristem, and it is always a sequential process. And all these events, that is conversion of vegetative to inflorescent meristem to floral, this is being called as something known as floral induction. And it is quite important that you should understand that the inflorescent meristem and floral meristem always come under the part of reproductive phase of development, while the vegetative SAM, as the name indicates, is a part of the vegetative phase of the flower development. Moving ahead, this is an important thing that I just discussed previously. I told you that the first set of or first identity of the SAM will be vegetative SAM. The vegetative SAM is mainly responsible for producing different part of the plant like the leaves and the stem. It has nothing to do with the formation of reproductive organs like the flowers and the fruits, etc. So the first thing that develops within an apical bud is a type of SAM which is called as the vegetative SAM, right? So we call it as what? The vegetative SAM or shoot apical meristem. The next thing is that this vegetative SAM changes its identity with the help of certain gene called as meristem identity gene. Please remember that the SAM will now undergo, or the cells under the SAM are actually undergoing some changes because a set of genes are activated. Those set of genes are known as meristem identity gene, and then the vegetative SAM gets converted into something called as the inflorescence SAM or shoot apical meristem. And this switch is mainly because of something called as the meristem identity gene, as clearly written in this figure, right? I hope that makes sense to everyone. So meristem identity genes are responsible for this switch. From inflorescent meristem, again meristem identity gene will result in floral meristem or floral SAM, and finally a flower will be developed. So a floral meristem is finally going to give rise to the flower. But conversion of floral meristem, so please remember vegetative SAM to inflorescence SAM to, if I say floral SAM, right? All these identity changes are controlled or governed by a set of genes called as identity genes. But please remember the conversion of the floral shoot apical meristem, that is the final identity, into a flower is the role of another set of genes which is called as floral organ identity genes, right? Which, what are they known as? Floral organ identity genes. So we have two main kinds of the genes which are responsible for floral induction. One of them is known as the inflorescence meristem identity gene, or sorry, they are known as meristem identity genes since they help in conversion of vegetative SAM to the inflorescence SAM to floral, and identity is changing, therefore identity genes or meristem identity gene. But conversion of floral SAM to flower is finally dependent upon something called as the floral organ identity genes. So two types of genes govern the floral development. That is the floral meristem identity gene and the floral organ identity genes. That is quite clear to everyone, right? Now we will discuss about each of these gene categories in detail, which will help us to solve all kinds of previous year CSIR questions, right? So let's discuss about the meristem identity genes. The meristem identity genes, they control the change in the SAM identity, that is from vegetative to inflorescence to floral. They are extremely crucial for activation of floral organ identity genes. Please remember that the meristem identity genes are always responsible for activation of the second class of genes called as organ identity genes. If some problem happens with the meristem identity genes, the organ identity genes will not be activated. So I hope that makes sense. So meristem identity genes control the change in identity and they are extremely crucial for activation of the next set of genes, that is organ identity genes. Now, the examples of meristem identity genes are a gene known as SOC1 or suppressor of constant 1. So here we are discussing the examples of something known as the meristem identity genes. I hope there is no doubt. So the first category of gene that we are about to discuss here is the meristem identity genes, right? So there is a gene which is known by the name of SOC1 or suppressor of constants 1. That gene activates a gene known as LEAFY or LFY. The LEAFY gene in turn activates AP1 or apetal gene. Please remember that the SOC1 and LEAFY, so the SOC1 and the LFY, they are master regulator for initiator of floral development. They are most crucial meristem identity gene for activation of flowering. You will understand why later on. Both LEAFY and AP1 are involved in a positive feedback loop. That means, so SOC1 activates LEAFY, LEAFY activates AP1, plus please understand that AP1 also activates the LEAFY gene. This kind of arrangement where one gene activates the other and the product of second gene further activates the first gene, that is called as a positive feedback loop, right? So three meristem identity genes discussed in front of you. SOC1 activates LEAFY, LEAFY activates AP1. LEAFY and AP1 are activating each other, which is called as a positive feedback loop, right? Please remember, once the AP1 gene is activated, the SAM gets committed to flowering, right? So that's very important. The shoot apical meristem gets committed to flowering. No matter what happens, if nutrients are available or not for the plant flowering or not, but the SAM is going to develop into a flower eventually, right? So once AP1 gene is activated, SAM becomes irreversibly committed to the floral development, right? Moving ahead, AP1 is both a meristem identity gene as well as a floral organ identity gene. So please remember, AP1 is a special connection. You will see that AP1 name appears as both meristem identity gene as well as the organ identity genes, which I will be discussing in the second half. Moving ahead, this next category of gene, which is floral organ identity genes. So floral organ identity gene, they code for proteins which are directly involved in whorl formation. That is the four whorls, the sepals, petals, then the stamen and the carpel. Please remember that most of the organ identity genes are homeotic in nature, right? That's a very important question, very famous question for CSIR. So meaning of homeotic is that their expression results in the formation of whorls at correct position. That means outermost whorl will always be calyx, then inside to it corolla, then you will have the androecium and then the gynoecium. That is correct order and correct position. If you mutate these genes, then the position of whorls get disrupted, right? The whorls may be formed at wrong position and such flower is not viable for reproduction further, right? So that is called as homeotic genes. Example of organ identity genes, I told you AP1 appears as a meristem identity gene also, and it appears in the list of organ identity genes also. So the example of organ identity genes are AP1, AP2, AP3, PI, and AG or agamus. So the next important point is that all plant homeotic genes, like the ones that we are discussing here, like I just said that AP1, AP2, AP3, and PI and AG, they are all homeotic genes, right? All of them contain a conserved nucleotide sequence domain called as the Matchbox domain, except the AP2, which is not containing that. Otherwise, all plant homeotic genes are containing a special conserved DNA binding domain called a Matchbox domain. So Matchbox codes for what? It codes for a special DNA binding Matchbox domain. This is a famous CSIR question also, right? So whatever appeared as a question ever, I have highlighted them with a different color, right? Moving ahead, role of organ identity gene in flowering is best understood by two kinds of model. One is known as ABC model and the other is known as ABCDE model. Somewhere you'll find it as ABCE model also, but that's not a problem. We'll discuss mainly the ABCDE model today and ABC. So let's talk about the ABC model of flowering. Now, this model uses Arabidopsis thaliana as its model, and you must be knowing that this is a diploid plant known as the model plant for most of the plant research. This model of flowering describes the interaction of organ identity genes. So how these 5 organ identity genes that I just mentioned, AP1 to agamus, right? How they are responsible for formation of the floral whorls. So 5 organ identity genes are there in Arabidopsis: AP1, AP2, AP3, PI, and agamus. And according to this model, they have been divided into 3 main classes, that is class A, B, and C. Moving ahead, let's discuss about this model. So this is very important. There are 3 main classes or gene types or gene activities according to this model. So class A activity or A class of gene, B class activity, and C class activity. Now under the A class activity, we have the AP1 and AP2. Under B class activity, we have AP3 and PI gene. And under C class activity, we have agamus gene. So what are the whorls that are being formed by these kind of activity or these genes? So the A alone activity, right, is responsible for formation of what? The formation of calyx or sepal. The A plus B is responsible for formation of what? The petal. So you can see this particular part, right? If you look carefully onto this particular segment here, this is called as what? A alone, right? So I will write here that this is A only, right? This is A only. So if any cell of SAM in which only A class genes are activated, that is destined to become a sepal. While any cell or any undifferentiated part where A plus B and both genes are activated, that is going to become a petal, right? Similarly, the section where B plus C genes will be activated, right? As you can see that this is the section where B and C both are involved, right? So where this part, if you mainly talk about this part, so we are trying to represent this particular section over here. So the section where both B and C are occurring or they get activated together, it results in formation of the androecium or the stamen. While the last part where C alone or C only, that is responsible for formation of the gynoecium or the carpel. So A alone gene activated, that part will become sepal. A plus B, that part is destined to become petal. B plus C, stamen. C alone or C only, then the gynoecium or the carpel. Let's have a look into this model in more detail from some research articles. This was a study by CSIR et al. Once again, this paper will be provided in my Telegram channel. It has been uploaded there. So absolutely no problem with that part. And if you look carefully into this thing, that's quite clear to us that the A alone activity is responsible for sepal. The part where A plus B are happening together, right? The section where A and B are there, that is for the petal or whorl number 2. The part where B and C are together is stamen, and the part where C alone is there, it's only carpel. But few other details are also important here. Please remember that the A and C class genes are antagonistic or cadastral in nature. Antagonistic means opposite. So what is the meaning of that? So please remember class A genes, right? Do you see that there is a clear-cut boundary between A and C, right? So A and C are never working together. B works with A, B works with C, but A and C are consider it as enemies of each other. So in whorl number 1 and 2, if you see here in whorl number 1 and 2, only class A genes are activated, right? So class A gene actually gets activated first, and if you look at this thing, the class A gene suppresses the class C genes in whorl number 1 and 2. That is quite clear. While class C gene suppresses the class C gene suppresses the A genes in whorl number in whorl number 3 and 4. You can see accordingly. So if you see here, so class A gene are not active or they are not working in whorl number 3 and 4. So whorl number 3 and 4 are stamen and carpel. Here, if you see this region, A genes suppressed. A genes are suppressed. And if you look at this region, here C genes are suppressed. So C genes are not working in whorl number 1 and 2, while A genes are not working in whorl number so A genes are not working in whorl number 3 and 4. That means A and C are enemies of each other or they're cadastral or opposite in nature, right? Looking at this thing, there are different kind of ABC mutant for which questions have been asked in the examination. So the wild type, everyone is clear with that it is a normal flower which will have sepals, then petals, then stamen and carpels. What will happen if there is an A function mutant? Suppose there are certain mutants generated in laboratory for which A genes are deleted. So if there are no A genes, then the entire region where the C gene was suppressed earlier by A gene, that will be actually showing the expression of C genes. So the first part can either be a leaf-like or calyx, leaf-like or the carpel, sorry. So CA is for carpel. Then the B and C is stamen, and then B and C is stamen, and then the last one is again carpel. So what we can see here is that in case of a function mutant, what has happened that the entire lower bar is now of the C, right? And B is already working with this thing. So if you look at this thing, there will be still four walls will be present, but the four walls are something like that. C alone was supposed to be carpel, C alone was supposed to be carpel, right? And then the B plus C was stamen and stamen. That's quite clear to us, right? What will happen if a B function is mutated? A and C will maintain their boundary, and it will be A alone, sepal, sepal, and C alone, carpel, carpel. C function mutant. So if C is absent, then the entire lower bar will be of the A, because the C you have deleted or it became mutated in natural form, but this part is of the B. So once again, we'll have sepal, petal, petal, and the sepal, right? Please remember, B function mutants are also known as indeterminate flower or flower within flower. Such questions have been asked in CSIR directly, that indeterminate flower will be formed by which kind of mutation. So the answer is B function mutation or B function mutant, right? Now, moving ahead, the next thing is to understand what will happen if all three genes are mutated, that is an ABC triple mutant. So in an ABC triple mutant, no whorl is being formed and all whorls are replaced by green leaf-like structure, right? And in biology or botany, there is a term given to this kind of flower, which is known as a pseudo flower, right? So it is known by the name of a pseudo flower. So ABC triple mutant, it is known by the name of a pseudo flower where all whorls are replaced by green leaf-like model. This is a version which is being developed as a modified version or enhanced model. So still we have A, B, and C. We have another class of gene called as E gene, and then one more gene called as D gene. The A plus E activity results in a sepal. A plus B plus E result in a petal. Then we have the B plus C plus E stamen. Then C plus E is the carpel. And then we have C, D, and E together, that results in ovule development, right? So it was A alone earlier, now it has become A plus E. It was A plus B earlier, so it has become now A plus B plus E, right? And it was B plus C earlier for stamen, so B plus C plus E. And it was C alone for carpel, it is C plus E. And finally, this section where we have what? C, D, and E. So this particular part together, C plus D plus E, results in something called as ovule development. So please remember, class E genes are known by the name of sepaletta genes or sep genes. That is, there are four types of sep genes discovered in plants: sep 1, 2, 3, and 4. And class D genes are known as seed stick genes or STK genes, right? Let's discuss the questions from previous year. So this is a question from 2011, part B. Which of the following whorls will be affected in an agamous mutant? So once again, this question can be easily answered from the figure that I just shown you in my lecture. So agamous mutant. So AG gene basically converts into what class of genes? The C class of gene. And if C class of genes are being mutated, so they are asking which whorls will be affected, right? So these C class of genes are basically involved in the whorl that we have, the carpel and the stamen. Therefore, the correct answer would be stamen and carpels, right? So that's quite clear. So agamous belong to class C, and if anything happens to agamous, it's a class C mutation, and problem can happen with stamen and carpel both. Next question is 2013, part B. Which of the following whorls will be affected in an AP3PI mutant? Once again, this can be answered from my lecture notes, this same figure. So AP3PI falls under the category of B class gene, and for B we have formation of petal and stamen. So that is B gene is playing role in both petal and stamen formation. So the petal and stamens can be impacted in a B class mutant or AP3PI. Now this is a part C CSIR question. They've asked you that which of the following statements are correct. More than one answers were correct. I will not be giving you options like PQR, PQRS. I want you to solve this question based on what I taught you during this lecture, right? So identify the correct statement regarding the flowering gene. So two main types of genes regulate floral development: meristem identity gene and floral organ identity gene. That's a correct statement if I remember. The important genes in Arabidopsis that play key role are AP1, leafy, and SOC1. This is also an important statement. If you remember that this particular genes, AP1, leafy, and SOC1, these were the meristem identity genes which I discussed with you, right? These were the meristem identity genes. So they do play a very important role in flower development, right? The gene that determine floral organ identity were discovered as floral homeotic mutants. Yes, correct. The floral organ identity genes, they are homeotic in nature, and that's how they were discovered. Most of the plant homeotic gene contain fat box gene. This is absolutely wrong. This was not fat box gene, but it was the Matchbox gene, which was discussed in my lecture. I will show you that what I taught you during my lecture. So there are two types of genes which regulate the floral development: meristem identity gene and organ identity gene. So that's why the statement number one is correct. Then important genes that regulate the development in Arabidopsis: AP1, leafy, and SOC1. If you remember, I told you that there is a flow chart which I just shown you that the AP1, leafy, and SOC1 are there, and then they were discovered as floral homeotic mutant. Fat box gene is a wrong statement, right? Moving ahead, 2014 June part C question. Identify the correct statement regarding flowering gene. Once again, we have to look at the correct answer. So this can be solved from the figure which I taught you. So activity A alone specifies sepals. So activity A alone specifies sepals. This is correct. Activity B alone specifies petal. No, petal is coming from activity of A plus B. So this is wrong. Activity of B plus C forms stamens. So B plus C forms stamens. Activity C alone forms carpels. Once again, this is correct. So only statement B is wrong here. So option A, C, and D are correct for this part C question. June 2015 question, a part B question. Identify the correct statement regarding leafy. Leafy is involved in floral meristem identity gene. I told you that it is a floral meristem identity gene. I never told you that it is involved in leaf expansion. It is not a root meristem identity gene, and it has no role with the far red light mediated action. So this is quite clear directly from my notes. CSIR June 2016 part C question. Identify the correct statement regarding flowering genes. So if you look at this question now, AP2 transcripts are expressed during sepal and petal development. We'll discuss it once again. You can see it can be solved from my figure. So AP2 transcript during sepal and petal development. So sepal and petal are involving something called as the A class gene. Let's see first, AP2 comes under which activity? So AP2 basically comes under what? A activity, right? And A activity is playing role in both sepal and petal development. So yes, AP2 transcript are expressed during both sepal and petal development. AG is considered as A class gene. This is wrong. AG is included in C class gene. AP1 is expressed during carpel development. So AP1 has nothing to do with carpel development. AP3 is expressed during sepal development. Once again, this is wrong. AP3 is class B gene. So let's have a look at this thing. So AP3 is a class B gene. So no role in sepal development because it is involved in the petal development. So the correct answer is option A in this particular case, right? These are the references, that two main papers that will help you. Please remember that all this information that I taught you here, the class notes, the solved question, and the study papers which I mentioned in the reference, they are available on my Telegram channel, for which the link is provided along with the video description. I expect that all of you will visit the Telegram channel and download all the information for free, right? So I hope that information helps for you. And let's target CSI December 2023 examination. Please let me know which video do you want me to upload next. According to the student demand, I will upload that content and with the previous year question. Thank you and have a nice day.